special event. And so I'll say a few remarks before I introduce our speaker. So this is the first annual Robert Millikan Cancer Epi Seminar. Our goal is each year to honor Bob's contributions to cancer epidemiology, especially breast cancer epidemiology, which is one of his passions and major areas of research and advocacy. And I think as all know, it's hard to fully express all of Bob's contributions to science, to school, to the department, his work with advocacy groups, and the many lives of the students, colleagues, and friends that that's okay. That friends that I can still barely read. Again, the many lives of the students, colleagues, and friends that he touched, and many of them are here today. And of course, it's now sort of a bit more over a year after his death, and I know a number of us remain sort of deeply affected by this. But I think it's important through this seminar and other events that we use this to honor Bob in a way that I think he would most appreciate through a discussion of the science that Bob loved so much, that he taught so effectively to so many people. So in fact, next year's event won't just be a seminar that next March, working with Bob's sister Anne, and others, that we're planning what will amount to, I think, a 3 or more day event, including a mini symposium, like a half-day symposium on breast cancer epidemiology that will invite UNC folks and outside folks to do a symposia. Anne is a composer, and she's working on the Millikan Symphony and so we'll have a concert, as well as the crew team that Bob was so involved in coaching. They every year have a Millikan Regatta, so we're planning a block of events to honor Bob through all the many things he was involved in: music, science, and the crew team. So I want to thank everybody who's donated to the Millikan Tribute Fund, and that's in part the source of support for this event, as well as the Lineberger Cancer Center, who has helped support the event. So with that, it gives me great pleasure to introduce this year's speaker, somebody that I think, somebody who's worked with Bob and has much to say about breast cancer epidemiology, particularly disparities, and that's Julie Palmer, who received her MPH from Boston University and her SED from epidemiology from Harvard in 1988. She's been on the faculty at Boston University since 1989 and is a professor of epidemiology at BU. And as many of you know, Julie's an international leader in women's health epidemiology of cancer, including breast cancer, through all of her publications, awards, appointments. One of her most important activities, and that's what had in part brought her and Bob together, and the Carolina Breast Cancer Study, is her work as the co-PI of the Black Women's Health Study, which is part of a consortium that includes CBCS, and Julie will talk about that consortium and some of the early results from the study. So I really want to thank Julie for being our inaugural speaker for this important event, and invite her to come on stage.
Thank you, Andy. Am I on the mic? Okay, great.
Okay, let me just
It's always wonderful to be at UNC, but it's a true honor to be asked to give this talk. You know, I, like you, love Bob and was a huge fan of his work, both before I knew him and afterwards. So it means the world to me to be here today giving this talk. This is sort of an overview, but I think I'm going to change it a little bit and leave out most of the second topic, which is giving some specific research from the Black Women's Health Study, which was just a small part of it anyway, because after meeting with students and other faculty over the last 24 hours, I've noticed a lot of interest in AMBER, and I've realized there's a lot to say about it, the consortium that we're all doing together. So I'm going to cut down that part, but I am going to start by telling you about the Black Women's Health Study, which is a cohort study that you know, many of you are probably not that familiar with. and talking about the reason for and the methods, and then I'll explain about the AMR Consortium, what that's all about, and then spend the rest of my time, as Andy said, talking about one of our specific analyses and that we're trying to understand. And again, this is something that Bob had a big interest in and wrote one of the seminal papers that raised the hypothesis that parity and lactation may have a differential effect for different subtypes of breast cancer. So the Black Women's Health Study, which began in 1995, was started because we noticed that there was a big gap in the research, and whereas women's health research had started picking up in the 70s and 80s, there was still very little population-based research being done on African Americans, and especially African American women. And yet African Americans were having the higher incidence and higher mortality from almost all of the chronic illnesses, including cancer. And as I think probably everyone in the room knows, mortality from breast cancer is higher, continues to be higher in African American women than white women. And even though the rates are dropping over time, there is this huge disparity, even though the incidence picture is a little bit different, which I'll get to later in the talk. So we decided to start a cohort study. We modeled it a lot on the Nurses' Health Study because both the overall PI, Lynn Rosenberg, and I had done our doctoral work at Harvard, so we were pretty closely familiar. In fact, Lynn was the first doctoral student to work on the Nurses' Health Study and write a thesis based on those data. So we were pretty influenced by it, and so if you'll note some of the methods are similar to that. The toughest part was figuring out a good source population for this cohort. We wanted to be able to enroll a large number of women. At that time, our goal was 50,000. We ended up with 59,000. And we needed women who had at least a high school education because the only feasible way to follow a large cohort is through written materials. And today we would be trying to do it by the internet. Back then we were using paper questionnaires, and not just paper questionnaires, but scannable questionnaires. And as we all know, it's not the easiest thing to fill those out, and we wanted people would answer the questions correctly. So we piloted using teachers, the list, and then you kind of need a list to get a cohort. You need some sort of base. So we tried with teachers, members of the National Education Association, which is teachers and teachers' aides. And education is a profession that African Americans have long been highly represented in. The other group we used was federal employees at the GS5 level and above, so that's clerical on up through administrative and scientific workers. And both of those gave us a decent response. It was about 16 or 17 response to a blind mailing, but the logistic problems were huge. And for example, the government employees, we would have had to go through every single little office of personnel. So we went to third source, which is magazine subscribers. So these are women who can read. We got the same response rate. The demographics looked about the same. So we ended up buying the mailing list, 400,000 names from Essence magazine, which at the time was the most popular women's magazine targeted towards African American women. And we figured with 400,000 we would get our 50,000. So we then picked states that would represent all regions of the mainland U.S. And that was pretty much it for the mailing. So we did this blind mailing to the 400,000 women. The other thing that was a key part of the methods was making sure our first questionnaire would be the hardest one they ever saw, because we wanted a lot of baseline information, and we also wanted people who would stay in the study over time. So we ended up with a 14-page OpScan questionnaire. Now we use Teleform, which is easier to, we can collect better data with Teleform. But at the time it was OpScan, so a lot of filling out bubbles. And that included a 6-page food frequency questionnaire. We sent it out, we got over 60,000 responses. Then we went through them. We decided not to include the very oldest women and not to include anyone who filled it out pretty poorly. So we ended up with a base cohort of 59,000 women and ages 21 to 69. In retrospect, we should have gone 25 to 69, or maybe even a tiny bit older. But we wanted to, the study is funded by the National Cancer Institute, so our primary aim was understanding the causes of cancer in black women, and to start off with breast cancer, since that's the most common cancer. But we wanted to be able to study cardiovascular disease and lots of other disease outcomes, and we wanted to be able to study early breast cancer, and we also wanted to be able to study reproductive outcomes. So that's why we did take young women, but women aged 21 to 25 move even more than everyone else. So I think 25 would have been a better starting point. So we do follow them every 2 years. We send out the 4-page. Once it was a 2-page, and sometimes it's been 6 pages, but it's no more than 6 pages, where we update information on disease occurrences. Well, we find out about new diseases. We have a list about 25 conditions and some specific cancers, and then we have a field for any cancer, and they write in the type. We ask about medication use, 6 or 7 major classes of medications, such as aspirin and NSAIDs, medications for diabetes, for hypertension, depression, and a few others. And we ask them to report any medications they have used for 2 days or more in the past few months, and then to write in the name of the medication. And then we get we update all sorts of risk factor data, and I'll get more to the types of variables we have. And I'm happy to say that we've managed to maintain a good response throughout this long follow-up. It practically kills us to do it. We mail the follow-up questionnaire. We get about 35% of participants send it back on that first mailing. So within 2 months, we get 20,000 questionnaires or something like that. And then we remail, we get another 15%, and after that, we get about 7% with each mailing. So we always do one priority mailing, so it goes in one of the big cardboard envelopes. It costs a lot more, but we get a better response from that. We mail about 7 times to non-responders. That's over a year and a half. By then, we're developing the next questionnaire. And we do some phone calling, but that's super expensive. But anyway, so with the 80% response, that's actually 80% complete response, so it includes deaths as well as people fill out the questionnaire. We know what's happening at 80%. We're happy with that in terms of the validity of the study. And this is what the participants looked like at baseline. The median age at baseline was 38. Now it's about 54, 55. They do come from all regions of the U.S. with large numbers in places like California, New York, Michigan, and certain states in the South. And you can see it's a pretty well-educated group. And you'll see later, very different from the Carolina breast cancer study. So we overrepresent black women of higher education. Although when you look across the U.S., in the birth cohorts who were in our study, 83% of African American women do have graduated from high school. So we get a lot of the women. We do not represent the women with the lowest education. And most of the women are non-Hispanic. We ask questions about the birthplace because we're interested in lots of factors, not so much genetic factors, which we can find out from genotyping, but more cultural factors that may be influenced by growing up, by what it's like being a black person growing up in the U.S. versus spending your early years in another country. So we do have people from other countries. And this slide just shows you the types of data that we've been getting. So it started off with the standard kinds of questions that are on any case control study that is looking at breast cancer. We have a full range of reproductive factors and medical history, family history, and the usual behaviors. And over time, we've added more questions that get at social and psychosocial factors that may influence not just other diseases, but cancer as well. I think we're learning more about how the whole stress pathway may influence cancer incidence as well as mortality. There's better data on mortality, but there is some for incidence. We've added some questions, hair relaxers, because so many black women do use hair relaxers. worried that, and there are a lot of toxic chemicals, but so far there's been no association with almost every outcome we've looked at, except for uterine fibroids. And we also asked about skin lotions that contain hormones, but that's it's hard to get information on that, so I don't think that's going to go anywhere. And then I talked about the SCS, and also we've mapped all of our addresses to Census Bureau data so we can download all those factors that tell us about the characteristics of the neighborhood. We also mapped to U.S. air pollution data. And we've, in the beginning, it was just a questionnaire-based study. In 2002, we were funded to collect DNA samples. That was in the days before Oragene kits, so we used the mouthwash swish method, which is good. We got some DNA from 50%, but it would have been better if it were Oragene because you get higher quality. Nevertheless, the samples seem to work and have been used and will be continued to use. And I don't have a slide, but finally 2 years ago, we were funded to collect blood samples from the cohort. And that was another reason it was good we had a young cohort, because even though we had already been following them for 14 or 15 years at the time, the median age was so young that most of the cancers hadn't occurred. And yeah, as you can see from this slide, this is maybe a year or two ago when we had data through 2011, we had lots of incident breast cancers out of the 59,000 women, but relatively few colon and lung, which you would expect to see high numbers if you had an older cohort. So that was good in terms of being late to collect blood samples, because it means we have our blood samples before so many of the cancers will occur. And there will still be a lot more breast cancers occurring too. So it's going to take us about 3 years to get blood samples from as many cohort members as we'll give it. Based on our pilot, we think we'll get 17,000 samples. And so initially they'll be useful for cross-sectional analyses, looking at biomarkers in relation to other things. So the study is, as I said, designed primarily for cancer. So we're very interested in these numbers of cancers, and we've been able to do some work on other cancers, but mostly on breast cancer. And we are also interested in other diseases. So the papers we've written so far have mostly been on breast cancer as far as cancers, but we've had 1 or 2 papers on lung cancer, one pooled analysis of pancreatic cancer mortality with the other cohort studies that have appreciable numbers of African Americans, a lot of papers on cancer screening and risk factors or correlates of obesity, and then other major areas. And if you look through this list, you can see most of these conditions, really with the exception of rheumatoid arthritis, have a higher incidence, and maybe lung cancer, have a higher incidence in blacks than whites in this country. So we're focusing in this cohort. There are other diseases that could be studied, but so far we're focusing on the ones that are both understudied and present a bigger health problem for African Americans. For example, sarcoidosis. I had never heard of it myself, and a lot of people hadn't, but our first questionnaire, so many women wrote that in under other medical conditions that we then added it to our next one. And out of our 59,000 women, 1,000 of them have sarcoidosis. And it's usually diagnosed by the 40s, 50s, so there won't be a lot of new cases. But that's part of the value of this study. So moving on to breast cancer in the Black Women's Health Study, again, it's a really questionnaire-based study. We can't do the kinds of work that's done in the CBCS, where you have everything right here and you can meet with the study participants in person and you can go to the hospitals and get the records. So this is a different type of study where everything is mostly done by mail and phone. So we rely on self-reports of cancer from the participants. Once they report a cancer, we then send them a release to allow us to get their medical records. Then we go to the hospital, PATH departments and medical records department, try to get as much information as we can. Some women don't want to give permission, surprise, surprise, and it's not everyone wants to just have their records accessed by other people. So For those, we go to the cancer registries, which itself is a big procedure, getting the cooperation of the cancer registries. But we're now into 18 of them, which represent most of the states that most of our people live in. So we can get good information on ER, PR status, now HER2 status, and then other tumor characteristics from either the medical records or the cancer registry. And so today we have ER, PR on 70% of our cases. Going forward, it will be on 95% or more. But we still have some cases from that were diagnosed in 1996, 97, 98, where we either couldn't get the information or they weren't tested for it, whatever. So we have older cases without the data, and plus this also includes our in situ cases, which were not, in the old days, weren't tested at all. Now they're more and more being tested. So in the future, we think we'll be getting this information on all of our cases. And if you're familiar with population data, you can see that the proportions are pretty much what you would see in SEER data for distribution in an African American population as far as ER positive versus ER negative. This slide, and it's a little bit hard to see, although it's easier to see here than in the paper. This is from a very recent paper by Clark and colleagues who looked at SEER data from the state of California. And it's a really great paper. I encourage you to read it. They were trying to look at whether the well-known age incidence crossover black-white disparity could be explained by subtypes. And I'm not saying they're the first people who asked this question, but they really explained it quite well. And so it's a little hard to see. The very first panel is luminal A, and the last panel is triple negative. And it's hard to see this first panel because the lines are so close together. But according to what they say in the paper, at every age, the incidence is higher in whites than in blacks. And what we see for the overall breast cancer is before age 40 or possibly age 45. If you just look at all breast cancers combined, looking at U.S. population data, you would see the incidence being higher in blacks than in whites. And then the crossover happens at that period, age 40 to 45. And at that point, overall breast cancer incidence is higher in whites than in blacks, and that continues through all the ages. What they're showing here is that if you just look at age incidence curves by subtype, you see for the largest group of ER-positive cancers, the first panel, that actually even at the youngest ages, the incidence was higher in the whites than the blacks. And then when you look at the last panel, that's easy to see with the naked eye, that at every age, incidence of triple negative is higher in blacks than in whites. Anyway, this is just a lead-in to why we are really interested in studying risk factors for triple negative breast cancer, even all, to the extent that we don't have information on triple negative, on all ER negative breast cancers, because it is, at most ages, it's twice as common in blacks as in whites, especially at the younger ages, it is twice as common. And the treatments are not as effective, and we don't know very much about risk factors for ER negative breast cancer. So that's a lot what our work in the Black Women's Health Study and work in the AMBER is designed for. And this is where I'm going to skip through just showing models, the Gale model, which is for white women, the CARE model, which was developed as a modifier of the, while I'm saying this, of the Gale model for black women, and these are the different factors included. I'll point out that the CARE model, which is for black women, only has 3 variables in: age at menarche, number of biopsies, and family history of breast cancer. It doesn't have parity or age at first birth. And when the CARE model was validated in Women's Health Initiative data, just from the 9,000 women in the Women's Health Initiative, with about 800 breast cancer cases, both the calibration and discriminatory accuracy were poorer than the Gale model for whites. We then published looking at how well the CARE model did in our follow-up data from Black Women's Health Study, but we looked not just at overall breast cancer, but we looked at how well it predicted the subtypes, and not surprisingly found that the concordance statistic was lower for ER negative. It's just a little bit higher than what you would find if there was no prediction at all, if it was 0.50. So one of the hypotheses, and again, this was initially raised in Bob's paper with his other colleagues from the Carolina Breast Cancer Study, and I see that Dr. Earp is in the back of the room, so shout out to you for the Carolina Breast Cancer Study. Published this landmark paper on the risk factors for the intrinsic subtypes, and in that paper, amongst other findings, was the very notable association that whereas for ER-positive breast cancer, Paris women had a reduced risk, that Paris women had an increased risk of basal-like breast cancer, and the first was luminal A breast cancer. They had an increased risk of basal-like breast cancer if they had a baby, and even higher if they had 3 or more babies. And most of the increase was just apparent among women who had not breastfed. So that set the stage for others of us to look at the same question, because that had not been seen or noticed or talked about before. Well, because most studies didn't have information on intrinsic subtypes or even enough ER/PR to look just by those 2 markers. So we then published similar findings in the Black Women's Health Study. And these are the questions we're trying to look at. And because each of our studies was provocative but not enough to really establish what is this all about? Is this real? Can we, are there recommendations that should be made based on the data? This was one of the reasons that we moved forward with establishing a collaboration, which we call the AMBER collaboration, which we worked together, Bob and I and Christine Amberzoni from Roswell Park Cancer Institute, met and worked together for over a year to put in a program project grant, because we felt it was a lot of work and more than just 1R1, and we wanted to be able to address multiple hypotheses, both genetic and non-genetic, and we wanted to be able to pool our 3 studies and possibly other large studies and be able to make use of establishing some cores so that the work would be shared, but with certain focuses done in different centers depending on their own expertise. And so, a brief overview, the 4 studies, you just heard about Black Women's Health Study. I'm going to assume you know about Carolina Breast Cancer Study. And Women's Circle of Health Study from Roswell Park is a case control study that was modeled closely on the Carolina Breast Cancer Study. They met with CBCS investigators. They got the questionnaire. Their questions aren't exactly the same. Some of them are exactly the same. Some are different. They had some additional questions. But the methods were very similar. It's a population-based case control study. They, like CBCS, they enrolled white participants and black participants so that they can do comparisons. And that study started much later. So at the time that we were putting in the application 3.5 years ago, they were still in the first data collection phase, and they're only now publishing their first papers from that study. And then the 4th study that is contributing data to this is the multi-ethnic cohort study, a cohort study that started just a year before the Black Women's Health Study out of Los Angeles and Hawaii. They have 5 ethnic groups, and altogether they had about 23,000 African American participants, women participants, and 12,000 men. So 2 cohort studies, 2 case control studies formed the basis for the data that would be used in AMBER. Okay, I promised I was meeting with students yesterday and we were talking about AMBER, and I said, oh, I have to go find this slide from one of my other talks, and I found it last night and stuck it in here. This is just to give you an idea of the scope of the AMBER. So really it's a 4-4-4 kind of thing, because there are 4 studies contributing data, there are 4 scientific projects, and there are 4 cores. And so you can see the projects, and the one is totally genomic, genetic susceptibility. Project 2, the work I'm presenting today, has to do with reproductive childbearing type factors. Project 3 has to do with energy balance, body size, and Project 4, vitamin D inflammation. And so each of those projects will have investigations based just on the questionnaire data, and then also investigations based on the genetic data, and finally looking at gene-environment interactions. So there are some pathways of interest, obvious interest to a certain project, like the estrogen metabolism pathway and Project 2. But then there's a lot of crossover of these pathways with the different projects. So that's what this kind of figure is trying to show. And then you see right in the middle is tumor heterogeneity. And the new thing and the really key thing about the AMBER Consortium is, and this is what we said to NIH, we need to look at, and not just all breast cancer, we need to look at subtypes, and we want to look at intrinsic subtypes. And to do that, we had to get tumor tissue data. CBCS was already getting tumor tissue. Black Women's Health Study hadn't started. Now we have it from 800 of our cases. And WCHS was just starting. And we want to have the reading, the IHC, and any other way we look at the tumor tissue done by the same people. So now there's a core at UNC that does all of the pathology work. We want all of our genotyping to be done the same way. And so we sent all our samples through a core at Roswell Park, and then sent the CIDR for genotyping. And we need harmonization of data. That's done at Boston University, and biostatistics, all of that. So that's the cores. But the tumor heterogeneity is this key part, because everything we look at, we're going to be looking at by subtype. That's our primary way of doing this. And then these 2 slides give you an overview of what we have. And there will be more cases. We've decided to wait and only update new cases once a year, or maybe every 2 years. So we're going to do it again in June. So this is our baseline data, and the first round of papers are being based on 2,450 ER-positive cases compared to all the controls, and 1,254 ER-negative, and 493 triple-negative, but actually we finally discovered there is some information from MEC, and we're just about to add 78 triple-negative cases from that. They don't have too many because they are the combination of oldest cases and earliest in time, so they just don't have the HER2 data for most of them. So the point I want to make, the reason I'm showing you ER positive, ER negative, we won't have information on intrinsic subtype until a year and a half or 2 years from now. Obviously, that takes a long time to do all the laboratory work, plus we've still been getting samples. So our first round of papers, we're just going to look primarily by hormone receptor status. And that's still valuable. We decided we didn't want to wait, that many more years to get some information. But the second round will be at least equally important. So we have large numbers. And this is just to kind of show you, we do have some early breast cancers, although I haven't broken it down by subtypes, but we know that ER negative are more likely to be younger. And the age distributions vary by each study. And then this tells you, this is really to show you across the studies how some of the factors that we were analyzing look. And there are differences for sure, especially between Black Women's Health Study and CBCS, and that has to do with the education differences mostly. So you can see that in CBCS, 47% of the participants have 3 or more births, whereas only 27% in Black Women's Health Study. And MEC is more like CBCS because both probably the population, but also they're the earliest birth cohorts when women were having more babies. And the same reason for the age at first birth, why MEC and CBCS have the earliest age at first birth. And what's really interesting, if you look across all 4 studies, do you see how few have a first birth after age 30? We do all of our cuts at age 25 because there aren't even that many with first birth after age 25. I mean, this is really different for anyone who's done breast cancer in epidemiology in white populations. You would be using very different category distributions. But here we have a very high proportion. The first birth is before age 20, and if not, then 20 to 24. Okay, I've kind of already talked about how we do the analysis, so I'm going to skip through this. But trust us that a lot of collaborative thinking has gone into this. We think we're doing valid analyses. And we, for the results I'm showing you, starting on the next slide, controlling for these other things made little difference. And we tried controlling in different ways. We really only had missing data for 2% for each variable, but we've used, I won't even say what we used because I'm at UNC, but we tried different methods, and they all came out the same. But we decided to actually control for all of those because reviewers are going to want some of the standard breast cancer risk factors. So here and here you can't even compare it, but in our first tables in the paper, we have just adjusted for age and cohort and time period. And then we do a multivariable odds ratio, and you can see how close they are. So this is the main table for looking at parity and lactation separately, and then I'm going to show you the combination of the two. And I circled that on the top just because there's so much on the table. And our big comparison is, what are we seeing something different for ER positive versus ER negative? And you can see we are. And we do see this increased risk of ER negative breast cancer in Paris women relative to nulliparous women. Then when we looked only among Paris women, so now the reference category becomes women with just one birth, we see there's a little bit of a trend towards increase with the largest number of births, and we did have enough to be looking at 4 more births. I guess we actually could go a little higher than that. And and we had very similar results for triple negative breast cancer. Looks a lot to me like ER negative. Whereas ER positive, there's no increase, and there's just a very small reduction observed here. I think less than a lot of people would expect to see. There is this common belief that ER positive breast cancer is reduced, risk is reduced in Paris women, and we don't see too much of a trend after controlling for other factors. This was a little different, it's 0.81 or 0.82 before we control for the other factors, especially age of first birth. And then for breastfeeding, we saw no association for ER-positive breast cancer and very small inverse association of lactation with ER-negative breast cancer and triple-negative breast cancer. We didn't have very good data on lactation duration. I mean, some of the studies asked detailed information on at least the first birth, and others didn't, so it was hard to harmonize. We did have a total duration variable for 3 of the studies, and when we looked at that in categories of total duration, we did not see any more stronger association for the women with the longest duration. In fact, it seemed, if anything, shorter duration was just as protective as that. And so, to me, this is the most interesting set of results, and especially looking at this for ER-negative breast cancer, which we're really interested in. So we're combining information on number of births and breastfeeding. And this table is among Paris women only. And so we have, the red line is women who did not breastfeed. The blue line is women who did breastfeed. And so you can see for either group, did not breastfeed or did breastfeed, risk goes up according to number of births relative to having only one birth. And the reference category, they have a common reference category, which is right here. So these are women who had one birth and did breastfeed. But in that group of women who, who did, even among that group of women who breastfed, there is a little bit of an increase. It's not, this is 1.3 maybe or something like that. It's not much, but there's a little hint of that. But it's really these women who did not breastfeed, even the ones who had one, they have a higher, at every level you see that red line is much higher than the blue line. And this is what was seen in that first CBCS 3 paper, and this is what we saw in Black Women's Health Study. And you can, it's a little hard to make out the confidence intervals because they overlap, but most of those estimates for the red line are the bottom limit is above 1.0. Whereas for ER-positive breast cancer, breastfeeding doesn't seem to make a difference, which is kind of, you would know that from looking at the overall result. But these lines are very close together, and if anything, in the women who never breastfed, there is a clear decrease in risk with increasing number of births. But actually, for both of them, there's this little bit of a reduction in risk for the most births. And try to understand better what's going on, we also looked according to age. We were wondering, well, is this just related to age, or is this just related to time since last birth? So first we looked at age, and we could see this was a stronger association in the youngest women for sure, but there is still evidence of an association in every age group. And again, this is where you wish you had bigger numbers, and we will have bigger numbers eventually. And we also look by age of first birth. So again, the bottom line is ER-negative breast cancer. And we saw just about the same association of parity with nullity parity, whether the first birth was before age 25 or after 25. This was our way of dealing with age of first birth, since we can't control for, there are some complicated ways now you could do some sort of controlling, but this is a good way to get rid of that factor to the extent that you're doing less than 25 and 25 plus. And we saw that each of those groups, we still saw an association of parity with ER-negative breast cancer. And then this is interesting to me because now in the women who had an early first birth, we do see this sort of expected inverse association of parity with ER-positive breast cancer, and it's only in the women with an early age at first birth and not in this group. So now to the question of time since last birth, and I'm going to show you a table and some figures, but I mostly have the table here because the figures don't have confidence intervals, and you might be wondering what the numbers look like. So again, we're focusing on ER-negative breast cancer. The reference category is women who did not have a baby, and then we took all the Paris women and categorized them according to how many years since their last birth. And, well, as expected, most of them would be in the longest time since last birth because most women who get breast cancer are older. And what's most interesting to me here is that we still see an association in the women who are a long time since their last birth, which suggests that what we're seeing for ER-negative breast cancer is possibly or probably different from whatever the pregnancy-associated breast cancer or the short-term adverse effects of pregnancy on breast cancer after a full-term pregnancy. We don't know. There are hypotheses about the biology, but I think this is important evidence to try to think about that. So we kind of see the odds ratios are sort of similar no matter what the category is. And for ER-positive, to the extent that any of these are different, we are seeing what you would expect to see, which is the longer experience the woman has of having had these pregnancies, these full-term pregnancies, the lower her risk. So now the figures, and so the first figure is just the top line is ER-negative breast cancer, and the other is ER-positive. And you can see that it's really just taking that table and sticking it into a figure. And so you can see that and these are odds ratios relative to nulliparous women. So you can see that the risk is increased for ER-positive at every level. And you can see that for ER-negative, and you can see for ER-positive, there's this little bit of a decline. We then thought, well, is there any way this could explain the time since last birth overall breast cancer effect that we've seen published in many studies, including the big Scandinavian studies? So we now put all the cases together and added a line to that. And so the green one in the middle is now all cases. And, you know, it's limited data, but what's interesting about this is now you see that kind of expected thing, although it doesn't go down very much here. But there's this short-term increase for the first 10 years after the birth, and then a reduction after that. But when you divide it into the two subtypes, you don't see that much of it. You don't see any increase for ER-positive. And you see an increase everywhere for ER-negative. And don't forget, in our population, 35 to 40% of cases, or at least 35%, are being classified as ER-negative. So we have fewer. So when we're looking at this together, only 65% are ER-positive. If someone were doing it in one of these Scandinavian cohorts, they'd have 86% or something. So this would probably be a little higher. And anyway, it's interesting, and I give Melissa some of the credit for raising this question. And we didn't set out specifically to address this subject, but once we got into it, we thought, you know, this is another piece we want to talk to people about. So those are all the data I'm going to show you, but if you have questions, there are, you know, of course, other things we did, so I might be able to speak to some of that. I think that the AMBER data, putting all of them together, it confirms the original findings. It does seem that there's an increased risk of ER-negative breast cancer and triple-negative breast cancer associated with being parous and with an increased number of births. And it does seem to be largely only in women who have not breastfed. And as I said, it persists with time since last birth. And given the differences in reproductive patterns of reproductive factors between the populations, this could explain some of the difference in the relative incidence of ER-negative breast cancer, ER-negative positive. Obviously, more research is needed. And I just want to thank the people from AMBER who are co-authors on this paper and have contributed to ideas for the analysis. And once again, to acknowledge Bob and his huge role in bringing this together and asking some of these important questions for our research. Thank you. Are there any questions? Sorry. Yes.
So the relationship between parity and ER-negative in women who didn't breastfeed is interesting. When you look at the number of births, if you've ever just looked at those two together, just whether maybe biologically what matters is ever having given birth without then